Welcome back to the deep dive. Today, we are not just talking about bugs. We are talking about architecture. We're looking at the structural logic of the most successful organisms on the planet.
It's a real shift in perspective, isn't it? I mean, usually when we talk about bacteria in a clinical sense, we talk about them as these invaders. We talk about the disease, the pathology.
Yeah, the enemy.
The enemy.
Yeah.
But today, using Chapter 12 of Murray's Medical Microbiology as our guide, we are going to look at them as engineers, as master builders.
And this isn't just, academic curiosity. The central premise here, and I think this is so important, is that if you understand how the machine is built, you understand how to break it.
That is the whole game. Exactly.
Yeah.
Antibiotics aren't magic spells. They are, for all intents and purposes, molecular wrenches that we throw into very specific gears in that machine.
So if you don't know where the gears are...
If you don't understand the gear, you don't understand the drug. It's the difference between memorizing a list and actually understanding the mechanism. Why does vancomycin kill staphylococcus? It bounces right off E. coli.
Right. Or why does a patient who's had their spleen removed suddenly become incredibly vulnerable to streptococcus pneumonia, but not, say, salmonella?
The answers are all in the architecture.
All of them.
So our mission today is to build a mental model of the prokaryotic cell, really from the ground up. We're going to map the territory. We'll start with how we classify them, the naming of the beast, so to speak.
The who's who.
Exactly. Then we'll go inside, look at the engine room in the cytoplasm. We'll spend a lot of time on the cell envelope because that's the battleground.
That's where the war is fought, absolutely.
And finally, we'll look at the external structures, the weaponry they use to interact with us.
We're essentially decoding the blueprint of life's simplest yet. I mean, you have to say, its most durable design.
Okay, let's start with that big picture. Classification. Murray opens this section with a metaphor that I think is really, really useful. He compares a bacterial colony to a city. It's a fantastic way to visualize it. When you're looking at a petri dish, you're not seeing a single organism. You are looking at a colony, a metropolis of, millions, maybe billions of identical clones.
A city of bacteria.
A city. And just like human cities have distinct characteristics, skylines, density, layout, even smells. Bacterial colonies do too. They have a macroscopic appearance that gives you your first set of clues.
So before we even get the microscope out, what are we looking for on that plate? What are these macroscopic clues?
Well, the most obvious one is color. Some bacteria are just show-offs. Staphylococcus aureus is named aureus because it means gold. It makes these beautiful golden colonies.
I've seen pictures of that. It's striking.
It is. Or Serratia marcescens, which produces this vivid red pigment called prodigiosin. But beyond just color, you're looking at texture and shape. Is the colony smooth? Is it rough? Is it mucoid?
Mucoid, meaning slimy.
Exactly, slimy. If you see a really wet, glistening, almost runny colony, that's a huge clue. It's practically screaming at you. This bug has a capsule.
Ah, so the city is covered in slime, which tells you something about its defenses.
Precisely. Then there's size, the margin of the colony. Is it a neat little circle or does it spread out in waves like Proteus does? This is all part of the initial characterization.
And then there's the smell. You mentioned this in our pre-show notes, this idea of olfactory diagnosis. Is that a real thing?
Oh, it is absolutely a real thing. Experienced microbiologists can often get a very strong hint about what's growing just by carefully opening the incubator. Pseudomonas aeruginosa. It has this unmistakable, sweet, grape-like, almost corn tortilla smell.
Which sounds pleasant, actually.
It is, until you realize what it means. But then you get something like Proteus mirabilis, which can smell like burnt chocolate or, honestly, rotting fish.
Not so pleasant.
Not at all. Or Eichinella corrodens, which famously smells like household bleach. These are all just metabolic byproducts, the exhaust fumes of these tiny cities being released into the air.
Speaking of metabolism, that's the next layer of classification, right? We don't just look at them. We see what they do, what they eat.
Right. This is biochemical profiling. We're basically giving the bacteria a buffet and seeing what they choose to eat.
Yeah.
We treat them like a patient and ask, what is your diet?
And a classic example of this is on McConkey agar.
The perfect example. The difference between a common gut bacteria like E. coli and a pathogen like salmonella often comes down to one simple sugar, lactose.
Milk sugar.
Milk sugar. McConkey agar has lactose in it and a pH indicator. E. coli loves lactose. It ferments it, produces acid, and the pH indicator turns the colony a bright bubblegum pink.
So pink means it's a lactose fermenter.
Right. Salmonella, on the other hand, it doesn't touch the stuff. It eats the other things in the agar and stays a boring, pale, colorless shade.
Hmm.
So with one plate, you've immediately split your potential suspects into two huge groups.
That's the macroscopic view, the city from an airplane. Now let's land the plane. We're going to the microscope. And this is the first major fork in the road, morphology.
The shape of the individual citizen. And while nature is, of course, incredibly diverse, for our purposes, medically relevant bacteria mostly fall into three geometric categories. You've got the spheres, which we call kochi.
From the Greek kokos, I think, which means berry.
Exactly, like a little berry. Then you have the rods, which we call bacilli. And finally, the ones that are spiral-shaped, like little snakes or corkscrews, things like treponema, which causes syphilis, or Borrelia, which causes Lyme disease.
But why the different shapes? I mean, is it just random evolutionary chance? Or is there a reason?
Oh, there is absolutely a reason. Form follows function, always. A sphere, a coccus, is the most robust shape. It has the lowest possible surface area to volume ratio.
Okay, what does that mean in practical terms?
It means it's the most resistant shape to drying out. It loses water the slowest. So what do you find on our dry, salty skin? Staphylococcus. A coccus. It's built for that environment.
In a rod.
A rod, a bacillus, has much more surface area. That's a disadvantage if you're trying not to dry out, but it's a huge advantage for absorbing nutrients. More surface area means more room for transport proteins.
No, it's a competitive environment like the gut where everyone is fighting for food.
Being a rod like E. coli is a great strategy. You can slurp up nutrients faster than your neighbors. It's an evolutionary trade-off.
That makes perfect sense. Now, they also arrange themselves in specific patterns. It's not just a random pile of spheres. There's a social logic to it.
And this is absolutely critical for diagnosis. If you look under the microscope and you see spheres, your very next question has to be, how are they arranged? Are they in a cluster, like a bunch of grapes?
Staphylococcus.
Staphylococcus. The name literally means bunch of grapes berry. They divide on multiple planes and stick together.
As opposed to chains.
Right. If they divide along a single axis and stay connected like a string of pearls, that's streptococcus. Streptos means twisted or pliable, like a chain. Seeing chains of cocci immediately makes you think of things like strep throat or necrotizing fasciitis.
And if they're in pairs?
Pairs are diplococci. And seeing diplococci should trigger a very short list in your brain. You're either thinking Streptococcus pneumonia or you're thinking Neisseria. The arrangement gives you a huge head start.
Okay, so we've got shape, we've got arrangement. But the single most important classifier, what we call the high-yield anchor, has to be the Gram stain.
Developed by Hans Christian Gram way back in 1884. It is still, to this day, the first and most important test you order when a sample hits the lab. It's foundational.
And it divides almost the entire bacterial world into two giant teams, purple and red, or I guess purple and pink.
Purple positive. That's the mnemonic you have to burn into your brain. If it holds onto that initial purple dye, it's Gram positive. If it loses the purple and takes up the pink counterstain, it's Gram negative.
Okay, I want to dig into the mechanism here because I think a lot of students just memorize the colors without really understanding the physics of it. Why does one stay purple and the other turn pink? What's actually happening on the slide?
It's a structural stress test. That's the best way to think about it. It's not just a dye, it's a process.
Yeah.
So, step one, you flood the slide with crystal violet. This is a small, water-soluble molecule that gets into everything.
So at this point, every single bacterium on the slide, regardless of type, is purple.
Everything is purple. Everyone joins the purple team initially.
Okay.
Step two, you add iodine. Now, iodine acts as a mordant. It's a chemical that helps the stain stick. The iodine gets inside the cells and binds to the crystal violet, forming these big, bulky crystal violet iodine complexes.
So we've gone from tiny little dye molecules to big, chunky purple crystals that are now trapped inside the cell wall.
That's the idea. They're much bigger and less soluble. Now comes the critical step, the differentiator.
Yeah.
The alcohol wash. You use acetone alcohol to decolorize the slide.
And this is where the two teams split up.
This is where they part ways. In gram-positive bacteria, which have this massive, thick, multi-layered peptidoglycan cell wall, think of it like a dense, cross-linked mesh. The alcohol hits this mesh and causes it to dehydrate. The pores in the mesh shrink.
It tightens up.
It clamps down. It effectively locks the door. Those big purple crystals that you formed, they're now physically trapped inside that shrunken mesh. They can't get out.
So they stay purple because the wall literally holds onto the dye.
Exactly. But now look at the gram negatives. Their peptidoglycan wall is paper thin. And, crucially, it's surrounded by an outer membrane made of lipids. What does alcohol do to lipids?
It dissolves them.
It dissolves them. So instead of locking the door, the alcohol just blows the outer membrane to pieces and punches huge holes in the thin peptidoglycan wall. All those big purple crystals, they just wash right out.
Leaving the cell completely clear.
Clear and invisible, which is why we need a final step. We add a counter stain, usually saffronin, which is red or pink. The now colorless gram-negatives pick up this pink dye just so we can see them under the microscope.
So, to summarize, gram-positive equals thick wall, which means it can trap the dye. Gram-negative equals thin wall and an outer membrane, which means the dye washes out.
Precisely. It's a beautiful and elegant test of fundamental structural differences. But, and this is where the exams love to live, there are the exceptions, the outlaws that don't play by the rules.
The ones that don't stain properly. Let's start with the big one, the mycobacteria.
Right, Mycobacterium tuberculosis. Structurally, it technically has a gram-positive type of wall, but it's different. It's covered in this incredibly thick, waxy coat of something called mycolic acid.
It's wearing a raincoat.
That is the perfect analogy. It's wearing a waxy raincoat. The crystal violet stain is water-based. It just beads up and rolls right off the wax. You cannot gram stain TB. It won't work.
So what do?
You have to use more aggressive stain, the acid-fast stain. It uses a strong dye called carbofuscin and heat to melt the wax and drive the stain in. Then, when you try to decolorize it with acid alcohol, the wax solidifies again and traps the stain. That's why it's acid-fast.
And the other major outlaw, the mycoplasma.
The mycoplasma. These are the nudists of the bacterial world. They have no cell wall at all, no peptidoglycan whatsoever.
So if there's no wall,
There's nothing to trap the dye. They don't stain with a gram stain. And clinically, this is a massive so what? If a bacterium has no cell wall, what happens if you treat it with penicillin?
well, penicillin works by attacking the cell wall, so
Yeah.
nothing happens.
Exactly. Mycoplasma is intrinsically, fundamentally resistant to all beta-lactam antibiotics because it simply lacks the target. You have to use a different class of drug, like a protein synthesis inhibitor.
That is such a key takeaway. The classification, the structure, it directly informs your treatment choices. Okay, let's move on from just naming and looking at them. Let's move inside the cell now, part 2. The cytoplasm, the engine room.
Right. So prokaryotes are largely defined by what they lack compared to us eukaryotes. They have no membrane-bound organelles, no mitochondria, no endoplasmic reticulum, no Golgi apparatus, and most importantly, no nucleus.
The DNA is just floating around in there.
It's concentrated in a region that we call the nucleoid, but it's crucial to remember it's not behind a wall. It's a single, circular, supercoiled chromosome, and it's directly accessible to the rest of the cell's machinery.
And this lack of a nuclear wall has a profound implication for speed, right?
It's everything. It's all about speed. Because they can do everything at once.
Yeah.
In our cells, we make RNA from our DNA in the nucleus. We have to process that RNA, add a cap and a tail, ship it out of the nucleus.
So the whole bureaucracy involved.
A huge bureaucracy. In bacteria, as the DNA is being read into a strand of RNA, ribosomes are already latching on to the beginning of that very same RNA strand and starting to make protein. Transcription and translation are coupled.
So they're building the machine before the blueprints are even fully printed.
That's a perfect way to put it. It's an assembly line with no waiting rooms. It allows them to respond to environmental changes almost instantly.
Now, floating alongside that main chromosome, you often find these other little bits of DNA, plasmids. I think of these as like downloadable content for a video game or cheat codes.
That's a fantastic analogy. Plasmids are small, circular, extrachromosomal pieces of DNA. They are not essential for the basic life of the bacteria. The bacteria can survive perfectly fine without them.
But they give it an edge.
They give it a huge edge. They carry the luxury genes, genes for things like antibiotic resistance, or for a particularly nasty toxin.
So a plasmid might carry the gene for a beta-lactamase enzyme, the thing that chews up penicillin.
Exactly. And the really scary part is that bacteria can share these plasmids. Through a process called conjugation, a bacterium can build a little bridge, a pilus to a neighbor, even a neighbor of a completely different species, and send over a copy of this plasmid.
So one resistant bacteria can essentially upgrade the entire local population.
In a matter of hours. This is horizontal gene transfer, and it's the primary engine driving the spread of antibiotic resistance in hospitals.
Okay, let's talk about the factory itself, the ribosome. This is where the proteins are actually built.
And this is the target for, I would say, at least half of the antibiotics we use. The magic here is that bacterial ribosomes are structurally different from our own.
This is the 70S versus 80S distinction.
Right. We measure ribosomes in Svedberg units, which is a measure of how fast they sediment in a centrifuge. It's related to size and shape, not just simple weight. Our ribosomes are 80S. Bacterial ribosomes are 70S.
And the 70S is made of two smaller parts.
Correct. The bacterial 70S ribosome is composed of a large 50S subunit and a small 30S subunit.
And for anyone doing the math at home, 30 plus 50 does not equal 70.
No, it doesn't. Svedberg units are not additive. It's just one of those things you have to accept. The numbers are 30, 50, and together they make 70.
But the key is that this structural difference is a perfect target.
It is the basis of selective toxicity. Drugs like aminoglycosides, gentamicin for example, or the tetracyclines, they bind specifically to the 30S subunit. They mess up the part that reads the RNA code.
And other drugs go for the 50S.
Exactly. The macrolides, like azithromycin or clindamycin, they bind to the 50S subunit. They block the tunnel where the new protein chain is supposed to come out. Either way, you jam the gears of the bacterial factory.
But because our ribosomes are a different shape and 80S structure, these drugs, for the most part, leave us alone.
For the most part. That is the holy grail of pharmacology, finding a target that the bug has, but we don't.
All right, let's move out from the cytoplasm to the main event. Part 3, the cell envelope. This is where Murray really spends the time, and so should we. We have these two competing architectural philosophies, the gram-positive and the gram-negative.
Or, as I like to think of them, the fortress versus the armored tank.
I like that. Let's start with the fortress, the gram-positive architecture.
So visualize a medieval castle. It has one single, massive, incredibly thick stone wall. That wall is peptidoglycan. In a gram-positive organism, this layer is enormous. It can be 150 to 500 angstroms thick. It's just layer after layer after layer of this cross-linked mesh.
It's a simple design, but it's incredibly strong.
Structurally simple, yes. But that thick wall needs to be reinforced and anchored. And threaded all the way through this thick peptidoglycan wall are these long anionic polymers. They're called teichoic acids and lipoic acids.
What's their function? Are they like rebar in concrete?
That is exactly what they are. They're the rebar. They provide rigidity and strength to the wall. The lipoic acids go all the way through and anchor the entire wall structure down into the cell membrane below it.
And clinically, why do we care about them?
Because they stick out from the surface. They are surface antigens. When our immune system sees lipoic acid, or LTA, it recognizes gram-positive invader. It triggers an inflammatory response.
It's a relatively weak response, right?
It's weak compared to what we're about to see with the gram-negatives, yes. It's a danger signal. It acts as a weak endotoxin, but it's more like a local alert, not a full-blown panic button.
Okay, so that's the gram-positive fortress. Thick peptidoglycan wall, reinforced with teichoic acids, and that's it. Nothing outside the wall. Now let's look at the gram-negative, the armored tank.
This is a much more sophisticated, much more complex piece of engineering. First off, the peptidoglycan layer, the actual wall, is pathetic. It's really thin, maybe only 1 or 2 layers thick. It's maybe only 5 to 10% of the total wall weight.
So if that's all they had, they'd be incredibly fragile. They'd just pop.
They would pop immediately under osmotic stress. But they have something the gram-positives don't have, and this is the defining feature: an outer membrane.
A second lipid bilayer?
A second lipid bilayer that sits completely outside the peptidoglycan layer. So the gram-negative envelope is a sandwich. You have the inner cytoplasmic membrane, then this thin peptidoglycan wall, and then the outer membrane.
And this outer membrane creates a sealed compartment between the two membranes.
Yes, the periplasmic space. It's like a DMZ, a demilitarized zone. And this space is absolutely crucial. It is packed with a gel-like matrix full of enzymes. This is where the bacteria keeps its digested juices, hydrolytic enzymes to break down nutrients, but it's also where it keeps its defense systems.
Such as beta-lactamases?
The perfect example. Beta-lactamases are enzymes that chew up penicillin. In a gram-negative bug, these enzymes are stored in the periplasm. So if you're a penicillin molecule trying to attack, you first have to get across the outer membrane.
Okay.
Then you find yourself in this periplasmic space where these beta-lactamase enzymes are waiting to ambush you and destroy you before you can even get to your target on the inner membrane. It's like a moat filled with crocodiles.
And getting across that outer membrane, the first barrier, isn't easy, is it? It's not just a normal membrane.
No, it is not. It has a very unique chemistry we'll get to. But even before that, just physically, it's a major permeability barrier. It blocks large molecules, and it blocks hydrophobic or greasy molecules.
This brings us to the pour-in problem.
Right. The bacteria has built this great armor, but it still needs to eat. It needs to bring in sugars and amino acids and other small nutrients from the outside world. So to solve this problem, it installs protein channels in the outer membrane called porins, gatekeepers. They're these barrel-shaped proteins that form a small, water-filled channel through the membrane. They act as a sieve. They will only let small, hydrophilic, water-loving molecules pass through.
But they block the big stuff.
They block the big stuff. And this right here explains the concept of intrinsic resistance. Take vancomycin. Vancomycin is a massive glycopeptide molecule. It's huge.
Right.
It kills gram-positives like Staph aureus very effectively because their peptidoglycan wall is completely exposed.
Yeah.
But if you try to use vancomycin against a gram-negative like E. coli, vancomycin is simply too fat to fit through the pore-in channel.
It literally bounces off the tank guard.
It bounces off. It can't even get to the moat, let alone the castle wall. So you would never, ever treat a standard E. coli or Pseudomonas infection with vancomycin. Not because they have a specific resistance gene, but because they are physically impermeable to it.
It's a structural mismatch, a design flaw in the drug from that perspective.
Precisely.
Now let's zoom in on the chemistry of that outer membrane. You mentioned the outer leaflet is special.
This is part 4, and this is absolutely critical. LPS, lipopolysaccharide. If you remember one single acronym from this entire deep dive, it has to be LPS.
And Murray, in the textbook, calls this endotoxin.
Because LPS is endotoxin, they are one and the same. It is the single most potent activator of the innate human immune system that we know of. It is an incredibly inflammatory molecule, and it makes up the entire outer surface of that gram-negative tank armor.
Okay, let's break down the structure of LPS. There are 3 components.
There are. First, buried deep in the outer membrane, you have lipid A, the L in LPS. This is the toxic part. This is the business end. This is the weapon. As long as the bacterial cell is alive and intact, lipid A is hidden away in the membrane.
But if the cell dies...
If the cell is killed, if it gets leg blazed by our immune system or by an antibiotic, it bursts open, and lipid A is released into the bloodstream.
And what happens when lipid A hits our system? What's the mechanism of the toxicity?
It binds to a specific receptor complex on our immune cells, especially our macrophages. The complex is TLR4, toll-like receptor 4, and a co-receptor called CD14. This binding event is like hitting a panic button. It triggers a massive, overwhelming signaling cascade inside the cell.
And the result of that cascade is?
The massive release of inflammatory cytokines. The 2 big ones are IL-1, interleukin-1, and TNF-alpha, tumor necrosis factor.
Because cytokine storm.
That is a cytokine storm. IL-1 acts on the hypothalamus in the brain to cause a high fever. TNF-alpha does all the other nasty stuff. It causes damage to the endothelial cells, lining our blood vessels, making them leaky. Fluid pours out into the tissues.
Which leads to a catastrophic drop in blood pressure.
Right. Widespread vasodilation and leakage. This is septic shock.
So ironically, if you have a patient with a massive gram-negative sepsis and you hit them hard with antibiotics, you can actually make them worse, at least initially.
It's a well-known phenomenon, sometimes called a Jerisch-Herxheimer reaction. You kill all the bugs at once, they all explode, they release a huge bolus of lipid A into the system, and the patient's blood pressure just tanks. You have to be ready for it with fluid resuscitation and vasopressors.
That's lipid A, the toxic anchor. What's next, moving outwards?
Attached to lipid A is the core polysaccharide. It's essentially a structural bridge that connects lipid A to the final component, which is the O antigen.
And this is a long chain of repeating sugars that sticks way out from the surface.
Exactly. It's waving around in the breeze, so to speak. And the key feature of the O antigen is that it is highly, highly variable. A single species like Salmonella can have over 2,000 different chemical versions of its O antigen.
Why? What's the strategic point of changing your surface coat so often?
It's about evading the police. Our adaptive immune system makes antibodies. If it makes an antibody that recognizes O antigen type 1, well, the bacteria can just switch to expressing type 2, and that antibody is now useless.
It's a disguise.
It's a constantly changing disguise. It's also how we do epidemiology. It's how we track outbreaks. When you hear about a food poisoning outbreak from E. coli O157H7, the O157 is telling you exactly which O antigen sugar chain that particular strain is wearing.
Before we leave LPS, there's a variant mentioned called LOS. What's that?
Right. Lipooligosaccharide. This is found in a few important pathogens, most notably Neisseria. It's basically a truncated version of LPS. It has lipid A, it has a core, but it lacks the long, repeating O antigen chains. It's short.
Does that make a difference?
A huge difference.
Yeah.
Because it's smaller and less well anchored in the membrane, Neisseria meningitidis is constantly shedding little bits of its outer membrane. It throws off these blebs of membrane that are just packed with lipid A.
So it's actively poisoning the host even while it's still alive and dividing.
Precisely. It doesn't have to die to release its toxin. This is one of the reasons why meningococemia, septic shock from Neisseria, is one of the most rapidly fatal infections in all of medicine. The endotoxin load in the blood is just enormous.
Incredible. Okay, this is a perfect transition. Part 5. We've looked at the wall structure, but now we need to understand how it's built. This is peptidoglycan synthesis. This might be the most technical part of the deep dive, but it's arguably the most high yield because this is where penicillin, vancomycin, and bacitracin all do their work.
We definitely need to slow down here and visualize the process. It's an assembly line. The goal is to build this strong, cross-linked mesh outside the cell. But you have to manufacture all the parts inside the cell, in the cytoplasm.
So, step 1, the cytoplasm. This is the brick factory. What are the bricks?
The fundamental building block of peptidoglycan is a disaccharide unit, 2 sugars linked together. NAM and NAG, N-acetylmuramic acid, and N-acetylglucosamine.
NAM and NA. Okay, those are the bricks.
Those are the bricks. But bricks alone don't make a strong wall. You need cement to hold them together. To make a mesh, you need to be able to link the rows of bricks sideways. So attached to every NAM sugar is a short chain of amino acids, a petapeptide chain, 5 amino acids.
And this is where the chemistry gets a little weird, a little unique to bacteria.
It really does. The chain ends with a very specific tail. 2 alanine amino acids back to back.
Yeah.
D-alanine, D-alanine. Now notice the D. In the human body and in almost all of life, amino acids are the L isomer, the L form. Bacteria, specifically in their cell wall, use D isomers.
Why? Why go to the trouble of using a different stereoisomer? Is it just to be different?
It's a brilliant defensive strategy. Our bodies, our tissues, are full of proteases enzymes designed to chop up and recycle proteins. But our proteases are exquisitely specific for L-amino acids. They can't recognize or bind to the D forms.
So by building their wall with D amino acids, the bacteria makes its wall completely resistant to our natural digestive enzymes.
That's it. It's brilliant, low-tech evolutionary engineering. Okay, so we have our unit, an NAM-NAG brick with this D-ala tail, but it's still inside the cell, in the cytoplasm. And it's a big charged molecule. It can't just float through the lipid cell membrane.
So it needs a ferry.
It needs a ferry. Step 2, the membrane crossing. The brick unit is attached to a massive 55-carbon lipid carrier molecule called bactoprenol. Think of bactoprenol as a crane or a conveyor belt. It's a greasy, hydrophobic molecule that lives in the membrane. It grabs the precursor on the inside, flips 180 degrees through the membrane, and presents it to the outside world.
And once it drops off its payload on the outside, the bactoprenol has to flip back inside to go get another one.
It has to be recycled. And that recycling step is a vulnerability. The antibiotic bacitracin, which is the active ingredient in Neosporin ointment, works by binding to the bactoprenol. It freezes the conveyor belt in place.
So the bricks pile up inside, but they can't get out to the construction site.
Exactly. If bactoprenol can't recycle, cell wall synthesis grinds to a halt.
Okay, step 3, the external assembly. We are now outside the cell membrane with our NAM-NAG units. What happens now?
2 things have to happen. First is transglycosylation. That's a fancy word for just connecting the sugars. An enzyme links the NAM of a new unit to the AN of the existing chain. NAM, NAG, NAM, NAG. This builds the long strands.
But long strands aren't a wall. They're just spaghetti. You need to link the strands together to make a net.
Right. You need a net, not just a pile of spaghetti.
Right.
And this is the final crucial step, transpeptidation, also known as cross-linking. You need to form a peptide bond to connect the amino acid chain of one strand to the amino acid chain of its neighbor.
And this critical job is done by a set of enzymes called transpeptidases.
Also known, famously, as penicillin-binding proteins, PBPs. And here's the mechanism that literally changed the world of medicine. The PBP enzyme is designed to recognize that D-ala tail we talked about. It binds to it, it snips off the very last D-ala to release energy, and it uses that energy to form the crosslink.
So the active site of the PBP enzyme is perfectly shaped to bind to D-ala-DL.
Perfectly. Enter penicillin. Beta-lactam antibiotics are molecular mimics. They are structural analogs. The beta-lactam ring at the core of the penicillin molecule looks, chemically and spatially, exactly like the D-ala bond. It's a perfect decoy.
So the PBP enzyme is going about its business. It sees the penicillin molecule, thinks, here's a wall piece I need to cross-link, and it grabs onto it.
And it's a trap. The beta-lactam ring is highly strained. When the enzyme binds, the ring snaps open and forms a permanent covalent bond to the active site of the enzyme. The enzyme is dead. It's totally and irreversibly inactivated.
It's like jamming a key into a lock and then snapping it off. The lock is permanently broken.
Perfect analogy. The PVP is permanently disabled. No more crosslinks can be formed. The wall gets progressively weaker as the cell tries to grow, and eventually the internal osmotic pressure causes the cell to lyse and explode.
It's a suicide substrate. The enzyme participates in its own destruction.
That's the technical term for it, yes.
Now, compare that mechanism to vancomycin. It also stops this same step, transpeptidation, but the mechanism is subtly and beautifully different.
It is. Vancomycin is not a decoy. It's a bouncer. It's a huge, bulky molecule. Instead of interacting with the enzyme at all, vancomycin binds directly to the substrate. It binds to the DLA tail itself. It forms a little cap, a little house, over the bricks.
So when the PVP enzyme, the transpeptidase, comes along to do its job,
It can't get to it. The vancomycin is physically in the way. It's a classic case of steric hindrance. The enzyme is perfectly fine, but it has nothing to work on because its target, the DLA, is covered by this giant vancomycin molecule, giving it a bear hug.
this distinction is so critical for exams. Penicillin kills the tool, the enzyme. Vancomycin hides the material, the DLA substrate.
You got it. And this also explains the mechanism of vancomycin resistance. How does VRSA, vancomycin-resistant Staph aureus, work? It's a single, tiny change. It mutates the gene for the pentapeptide so that the tail is now DLA-lactate.
It swaps one molecule, an oxygen for a nitrogen.
That's all it takes. That one atomic change is enough. The vancomycin molecule can no longer bind. The bear hug fails. The PBP can now access the tail and build the wall, and the bacteria survives in the face of what should be a last resort antibiotic.
That is high-yield gold right there. Okay, we have painstakingly built the wall. Let's finish by looking at the structures on the exterior, part 6. The weaponry and appendages. Let's start with the capsule.
The capsule. This is a gelatinous, usually polysaccharide layer that coats the entire outside of the cell. I like to call it the slippery suit or the invisibility cloak.
How does it hide the bacteria from the immune system?
Well, one of the main ways our immune system clears bacteria is through a process called opsonization. We coat the bacteria with proteins, either antibodies or a complement protein called C3b that act like little handles.
They make the bacteria tasty for phagocytes, or white blood cells.
Exactly. They make it easier to grab. A capsule is a thick, slimy layer that covers up all those opsonins, all those handles. The phagocyte tries to grab the bacteria, but it's like trying to grab a greased watermelon. It just slips away.
So it's antiphagocytic.
Purely antiphagocytic. And this is the #1 primary virulence factor for some of our most dangerous pathogens. Streptococcus pneumonia, Haemophilus influenzae type B, and Neisseria meningitis.
And this directly connects to the spleen. Why is the spleen so important for dealing with these specific bugs?
The spleen is a highly specialized organ. Its unique blood flow architecture, the sinusoidal system, is designed to slow down blood and force it through narrow spaces packed with macrophages. This is the one place in the body where we can efficiently trap and clear these slippery, encapsulated organisms from the bloodstream.
So if you don't have a spleen, if you're a splenic from sickle cell disease or a trauma,
You lose that specialized filter. You become massively susceptible to overwhelming sepsis, specifically from these encapsulated bacteria. It's why those specific vaccines are non-negotiable for any splenic patient.
Now, you said capsules are usually polysaccharide. Is there an exception?
There is always an exception for the exams. Most are made of sugar, but Bacillus anthracis, the agent of anthrax, is different.
The rebel.
The rebel. Anthrax has a capsule made of protein, specifically poly-D-glutamate. It serves the exact same antiphagocytic function, but it's chemically unique.
Okay, let's move from defense to offense, or at least movement. Flagella.
The propellers. These are long, whip-like tails made of a protein called flagellin. They are incredible little machines. They have a basal body that acts as a rotary motor embedded in the cell membrane. There's a rotor, a stator, a drive shaft. They can spin at thousands of RPM.
And they're not powered by ATP, which is what powers most things in a cell.
No, which is fascinating. They are powered by the proton motive force. The flow of hydrogen ions, protons, across the cell membrane is what drives the turbine. It's like a microscopic hydroelectric dam.
And the movement isn't just random swimming, it's purposeful, this run and tumble motion.
Right, it's a biased random walk. When all the flagella motors spin counterclockwise, the flagella bundle together into a sort of super propeller, and the bacteria shoots forward in a straight line. That's a run.
And the tumble.
When the motors reverse and spin clockwise, the bundle flies apart, and the bacteria just sort of chaotically flips around in place. A tumble. By controlling the length of the runs and the frequency of the tumbles, the bacteria can gradually move toward a food source or away from a poison. It's called chemotaxis.
And clinically, the flagella protein itself is an antigen. It's the H antigen.
Yes, highly antigenic. It's the H in E. coli, O157, H7. The O antigen is from the LPS. The H antigen is from the flagella.
Lastly, for external structures, the pili or fimbriae.
I call these Velcro. They are much shorter, thinner, hair-like bristles that cover the surface of the bacteria. Their job is not movement. Their job is adherence,
Sticking to things.
sticking to things. They have spiralized proteins on their tips called adhesins or lectins that bind with high specificity to certain sugars on the surface of human cells.
And the classic example of this in action is a urinary tract infection, a UTI.
Absolutely.
Hmm.
E. coli causes about 80% of all UTIs. Why? Because it has specialized pili, called pili, that bind specifically to receptors on the uroepithelial cells lining your bladder and ureters. The constant downward flow of urine is strong, but the E. coli uses its pili to hold on like a burr on a sweater.
And there's one very specialized type of pilus, the sex pilus.
The conduit for, not love, but at least for genetic exchange. It's a longer, thicker pilus that initiates the formation of that conjugation bridge we talked about earlier to transfer plasmids carrying antibiotic resistance genes.
All right, part 7. Let's quickly touch on 2 specialized survival structures before we wrap up. We have to mention spores.
The endospore. This is the ultimate survival bunker. It's a dormant, non-reproductive structure, and it's really only made by a few medically important genera of gram-positives, specifically Bacillus and Clostridium.
Which are the gram-positive rods of doom. Anthrax, tetanus, botulism, gas gangrene, C. diff.
All the heavy hitters. When these bugs sense that they're running out of food or the environment is becoming hostile, they don't just die. They hit the panic button, they replicate their chromosome, wrap it in multiple thick keratin-like protein coats, and then they completely dehydrate the core.
And they saturate it with a chemical, calcium dipicolinate.
That's the chemical signature of a spore. The calcium dipicolinate complexes with the DNA and makes it incredibly resistant to heat, to UV radiation, to desiccation. A spore is metabolically inert. It's basically a tiny armored rock.
And this is why they are so hard to kill.
So hard to kill. A spore can survive boiling water. It can survive treatment with alcohol-based disinfectants. This is why, for a patient with C. diff, you must wash your hands with soap and water. The alcohol gel does nothing to a spore. It just moves it around on your hand.
Whereas soap and water mechanically removes them. That is a truly practice-changing detail.
It is. The structure dictates the protocol.
We have covered a massive amount of ground today. We've built the city, we've reinforced the fortress, we've armored the tank, we've detailed the assembly line, and we've examined all the external weaponry.
It really is a comprehensive model. And if you can visualize these structures in your mind's eye, the thick purple mesh of a gram-positive, the outer membrane sandwich of a gram-negative, the PBP enzyme acting like a little construction worker building the wall clinical microbiology, stops being about memorizing endless lists of facts, and it starts being about logic.
Before we hit the rapid recall questions, I want to pose that provocative thought we hinted at in the beginning. We made a big deal about how ribosomes are a great antibiotic target because bacteria have 70S ribosomes and we have 80S ribosomes. But there is a ghost in our own machine.
The mitochondria.
Our mitochondria, the power plants in every one of our cells.
Yeah.
But evolutionary theory, the endosymbiotic theory, says the mitochondria used to be free-living bacteria that got swallowed up by an early eukaryotic cell billions of years ago.
And the evidence for this is incredibly strong. Mitochondria have their own DNA, and it's circular, just like a bacterium's. They divide by binary fission, and they have their own ribosomes.
And what size are those mitochondrial ribosomes?
They are 70S-like. They are structurally very, very similar to bacterial ribosomes.
So the question is, when we take an antibiotic like gentamicin or chloramphenicol to kill a bacterial infection, are we accidentally bombing our own mitochondria at the same time?
We are. And this is thought to be the mechanism for some of the specific toxicities we see with these drugs. Chloramphenicol, for example, can cause bone marrow suppression. Why? Because it's a potent inhibitor of mitochondrial protein synthesis, and your rapidly dividing bone marrow cells are incredibly dependent on their mitochondria.
So the side effects are a case of friendly fire against our own ancient bacterial tenants.
Exactly. We are, in a very real sense, part bacteria, the humbling thought.
It really is. All right, let's solidify all of this knowledge. It's time for the rapid recall.
Let's do it. I'm ready.
Okay, listeners, I'm going to give you a clinical scenario. I want you to try and identify the answer and, more importantly, the why before our expert does. Question 1. You are treating a patient with suspected meningitis. The lab calls and reports gram-negative diplococci from the cerebrospinal fluid. A few hours later, the patient suddenly crashes with profound hypotension and develops a widespread petechial rash. Which specific molecular component of the organism is most directly responsible for this rapid septic shock?
Okay, the answer there is lipid A, which is the toxic component of the LOS or LPS. Why? The organism is almost certainly Neisseria meningitis, the classic gram-negative diplococci causing meningitis. Gram-negatives have lipopolysaccharide, or in this case, liposaccharide LOS, in their outer membrane. Lipid A is the part that binds to TLR4 on our immune cells, triggering the massive cytokine storm with TNF-alpha and IL-1 that leads to fever, vascular leakage, and shock. Question 2.
A pharmaceutical company develops a novel antibiotic that works by blocking the function of bacterial pyrophosphate phosphatase. Which specific step of peptidoglycan synthesis would be inhibited first?
The answer is the transport of the NAMAG precursor units across the cell membrane from the cytoplasm to the outside. Why? Bacterial is that lipid carrier, the conveyor belt, that flips the cell wall building blocks from the inside to the outside. The phosphatase step is required to recycle it. Inhibiting it, just like the drug bacitracin does, traps the precursors inside the cell. The construction site on the outside runs out of materials. Question 3.
This one is a bit of a trick question about technique. You are performing a gram stain on a sample you know contains Staphylococcus aureus. However, you're distracted and you accidentally forget to add the iodine step. You perform all the other steps correctly: crystal violet, the alcohol wash, and the safranin counterstain. What color will the Staphylococcus aureus be at the end of this botched procedure?
That's a great question. The answer is they will be pink or red. Why? This tests your understanding of the mechanism. The iodine is the mordant. Its job is to form large complexes with the crystal violet that get trapped in the thick peptidoglycan. Without the iodine, the crystal violet molecules remain small. So when you add the alcohol, even though it's a gram-positive wall, the small dye molecules will just wash right out. The cells will become colorless and will then pick up the pink safranin counterstain. Question 4.
A 19-year-old college student presents with a persistent cough and is diagnosed with walking pneumonia caused by mycoplasma pneumonia. Explain, based on its structure, why it would be completely ineffective to treat this patient with ceftriaxone, a powerful cephalosporin antibiotic.
The answer is because mycoplasma lacks a cell wall and therefore lacks peptidoglycan. Why? All beta-lactam antibiotics, which includes penicillins and cephalosporins, work by inhibiting the transpeptidase enzymes, PUBPs, that build the peptidoglycan cell wall. Since mycoplasma is one of the outlaws and has no cell wall at all, the drug simply has no target. It would be like trying to sink a ship that was never built. Question 5.
A microbiologist identifies a bacterium from an environmental sample. It's a large gram-positive rod that forms spores. On further analysis, it's found to possess a specialized capsule made not of polysaccharide, but of a polypeptide of polydeglutamate. What is the likely genus and species of this organism?
That unique capsule is a dead giveaway. The answer is Bacillus anthracis. Why? It is the classic exception to the rule that bacterial capsules are made of sugar. The anthrax capsule is protein, which is highly unusual and a key virulence factor. Question 6.
You are studying a strain of E. coli that is highly resistant to a broad-spectrum penicillin. You run a test and find that the bacteria does not produce any beta-lactamase enzymes. Furthermore, its penicillin-binding proteins are completely normal and susceptible to the drug. What is the most likely structural mechanism of this resistance?
The answer must be a mutation in the porin channels in the outer membrane. Why? For a penicillin to work on a gram-negative like E. coli, it has to first get into the periplasmic space. It does this by passing through porin channels. If the bacteria mutates its porins to make the channels smaller or to change their charge, it can physically exclude the antibiotic molecule. The drug can't reach its PBP target because it's blocked at the outer gate. This is a major mechanism of resistance via impermeability.
And there we have it, the fundamental logic of the invisible world made visible.
Master Chapter 12 of Murray. Seriously, read it alongside this. It's the foundation for every single bacterial pathogen we're going to discuss from here on out.
Thanks for listening to The Deep Dive. We'll see you next time.
Stay curious.